To the wonderful organizers of this meeting, and of course, BiGEvo and ISCB that they've really been, very, tolerant of our doing everything last minute and getting money last minute and many things last minute. So thank you, everybody. And of course, I have to thank CiÄ as a sponsor of this meeting. And of course, everybody that collaborated in BiGEvo. And now I'll tell you about the things that are of interest in my lab, which is some of what you said before about microbial communities. What do they really do, and how can we ever understand their complexity? And the communities that I have studied are mainly from Cuatro CiÄ. And this one. And because I am always, running at the end, I always start with thanking people. And these particular projects were first funded by, the CONACYT project on Fronteras, which was a collaboration with Adán Fuentes, Jorge Rocha, Gabriel Moreno, and Maribel. And then later, we were also fortunate to have a grant, which is a joint effort from CiÄ and the University of California, between Maribel and Alejandra and Adam Ocean, here they are, and me. And then we also had a collaboration from Brazil, and we've also had collaboration for sequencing, many of the genomes from Solena. And of course, I have to thank everybody in my lab, students and postdocs and lots of undergraduate students, and a lot of other collaborators, current and from other years, like Mike, Valeria, Luis, and other people that are here, like Jesus Silva, who said, from the Health Institute, who really helped us along understanding all the antibiotic things that we were doing. So I'll talk first about natural communities, NACs and microbialites. And the theme of this talk is microbial resistance to antibiotics. These are antibiotics used in the clinic, and what they are a big problem right now, and resistance to antibiotics in pathogen strains is a big problem. And there are a lot of resistances, and there are a lot of different discourses on how these came about, how these super resistant bugs appeared, and how they dispersed. So that's one of the things that I'm going to take this approach of going as far as possible from a clinical model to try to understand the story of the evolution of microbial resistance, and to really go very far from the clinic. And then I'll show you how resistant patterns are shaped by ecological structure, by the taxonomic composition and stratification of the different bacteria. And then we also take the advantage of the synthetic community that we have been studying for ages, and that we have a lot of information about their interactions and their genomes, and it's been a great model to be able to study this. So what I'll tell you just is like, antibiotic resistance, it's really a big problem, and now there's a concept that is called One Health, in which the idea is to see this as a larger problem, because antibiotics goes beyond the clinic, and you will see it in effluents, and you will see it in many places. But also this clinical view that is still very, very persistent is that it's a result only of the abuse of the clinical antibiotics in hospitals. And also there's another focus, which is what they are called escape strains. Of course, those are pathogen strains that are very worrisome, that appear in hospitals. And most of the things that we know about antibiotic resistance comes from these escape things. And the clinical view is that antibiotics are, the resistance is the result of the use. So here we see on the top, when an antibiotic comes to the market, and then on the bottom, when the resistance was first observed. And so that is the story. You start using an antibiotic, and of course the strains, become resistant, so they cannot use that antibiotic any longer. So that's been a little bit the history about the clinic, of what some things you can keep using, some others you have to move on. And another thing is that antibiotic resistance, can spread, not just because the bug itself, moves, like COVID, where you move it around, but also because they're horizontally transferred plasmids. So within some taxonomy groups, this is moving around, and you would be then seeing the same bug that appeared once in a place. Then 10 years later, you see it, everywhere in the world. So that's sort of one of the discourses about antibiotics. The thing is that about antibiotics, and in general about bacteria, this is the main set of bacteria that have been studied in literature. For anything that you think about, there is, these 10 species are in 50% of all the papers that you would read. And so most of the things that we know it from a very small set of bacteria. Some of these are called escape. Escape come from E. coli, Staphylococcus, Klebsiella, Enterococcus, and Acinetobacter. So those, for those we know a lot of things about antibiotic resistance. But why we went to the mats, and we went to the microbialites, and we went to insect and studied these bacteria that have not been impacted by clinical antibiotics. So this is a different story now. So the story is to bring ecological and evolutionary perspective to antibiotic resistance. The idea is to go beyond escape bacteria, and the idea is to go back millions of years ago, before there were even humans to infect. And that is very easy in Cuatro CiÄ negas, because we have a lot of places that come from early before we had plants and animals on Earth. And the idea is to have analog systems of things that existed a long time ago, and work with those groups. And the story that I want to tell you is that a bacteria cannot produce an antibiotic if it cannot resist it itself. So my idea is the second that an antibiotic is synthesized in bacteria, there is also resistance there. Okay? All bacteria are resistant to their own antibiotics. And this resistance can move, or can be evolves and in another strain. Because all of this is happening all the time in microbial communities. So all the time that a new antibiotic is around, a new resistance emerges. But it happens, it happened a long time ago, and it was all very quickly. And the types of resistance, are basically of three forms. An antibiotic gets into the cell, and you immediately take it out, right? You have a pump, that is like if you have a clog, you immediately get rid of the water. So you have an antibiotic, you immediately get rid of that antibiotic with efflux pumps. Otherwise, you can do other things. If you have an antibiotic, you can have a special enzyme to modify the antibiotic. You can clean the antibiotic by basically getting, you chemically can transform the antibiotic. And so that's another way to get rid of an antibiotic, like inactivation. And then you can also just become resistant to the antibiotic itself by modifying the target. The target can be mutated. If it's a protein, a few amino acid changes can make it resistant. That would be intrinsic resistance. So those are mainly the three ways that bacteria can go out this thing. And this is happening all the time in communities. In these communities, they are competing. Maybe one bacteria is killing another one, or trying to kill it with an antibiotic. Or maybe there's another one that is, making an enzyme to inactivate the antibiotic. So that's very much, the game that we have here. And I'll show you what we have of this game, what we understand now from the Turing set, from the schematolite, and from the microbial mass. So part one let's go to see what happens in stromatolite and mats communities. So suppose you take a sample and put it on a petri dish in the presence of an antibiotic. With some antibiotics, you will have just tons of growth. So most of the bacteria are basically resistant, in this case, to vancomycin. But you will see something else. You will see that some bacteria are not allowing other bacteria to grow, around. So they are just helping me to show you how this is so common. How bacteria are making their own antibiotics all the time and are inhibiting other bacteria that are around them. And we see that every time we plate. And if we collect this bacteria, now we have a bacteria that is antagonist for another one. And so it's very easy to find. Now, what if I take bacteria from these sites, and I will plate it in every antibiotic that I can? I will plate this on vancomycin, carbonicillin, and many different antibiotics. So on the top, we have antibiotics that inherit the cell wall synthesis. And what we have is this is the stromatolite sample, and this is the mat sample. Both samples are full of bacteria that are resistant to all of these antibiotics that I'm not going to tell you, rifampicin, erythromycin, streptomycin, tetracycline, chloraphenicol, all of this. And look at vancomycin in the middle. You have all this resistance in the stromatolite, and you have some resistance, not that much, in the mat. For me, that I'm a microbiologist and I know how vancomycin acts, if vancomycin is killing something, I know there are more gram-positives there. So it's like, there might be a different taxonomic composition. So Gerardo did this work, and Gerardo took some of these sample of strains, and he sequenced the 16S of many of those to see who they were. And now what we have is on the stromatolite, we have a sample that are mainly gram-negative. That's why those are more resistant to some antibiotics or other. And then on the other one, we have that sample is more enriched to gram-positive. This is a 16S of randomly taking bacteria from the different antibiotic-resistant samples. One other thing that I want you to notice, if you see this block here, it means that these bacteria are resistant to everything that we tested. That's the multi-resistance. That's what we see in the clinic. And we didn't use anything in the from the clinic, right? So this is happening all the time. This multi-resistance is already there, okay? And, we have just different taxonomic groups in each of these. Part 2. What if I take a sample that I know really well and I know how they compete with each other? So we have such a sample, we call it the bars collection. Our bars collection are bacillota that are antagonist, resistant, or sensitive. If I do a phylogenetic mapping of this, I see that these are all the resistant, all the orange. They are resistant and they are antagonist. All the blue ones are resistant, and the rest are sensitive. Sensitive to whom? To the attack from the orange one. So this is a sample where I know that they interact. I know that the orange kills everybody it can from the sensitive group. I know that nobody kills these resistant bacteria, but much more, I know that these resistant bacteria, if in the presence of this community, are going to help them out and are going to neutralize the antagonist. They are not going to let all this killing happen, right? These are our police, okay? So the other thing that is interesting about is the microdiversity. These plates are within the same species, and within the same species, then what we have is different strains. And these different strains, they are not identical. That's something that Emmanuel in my lab has been exploring. He's exploring what does it mean, all this redundancy? Is that the same as equivalence? Can we just interchange one strain for the other one? No, you cannot interchange them, but they do have similar ecological goals. So you can now take one by itself. You don't need them to have in a community, And you can have a plate with your bacteria, and you will see that you can kill them. So basically, this has been our model system. We have a lot of interactions. We know that from the pangenome that they are not identical, and although they are similar, so we understand the microdiversity of the strains. And we know also what is their potential to make these what are called BGS, which are biosynthetic clusters. That's how bacteria make their antibiotics. So we want to know, how do they make their antibiotics? How do they make their antidote, the enzymes to resist? So they make their antibiotics with these kind of things. But what I think is interesting, if I look at all the clusters for antibiotic synthesis or other kinds of toxins, what I see is that each taxonomic group is very different from the other. They are not like, there's no horizontal gene transfer and everything is everywhere. It's very cohesive and congruent by taxonomy. And so by every taxonomic group, like this one, the antagonist, it has more antagonist systems than the other ones, but also these antagonist genes are particular to its group. And resistant and antagonist have the most VGCs. If I use a system to map them to resistance from humans, I find all these similarities. Everything that something has color, it means that we found a match in our sample. But I see again that they have a particular cohesiveness by plate. They are just not randomly distributed. And if I look at what this has to do with inactivation or the efflux or whatever, there are different strategies, like Bacillus cereus that you just saw Roles seeing. This Bacillus cereus has this strategy. I'm going to inactivate everything. It's not altering its target, and it's not being able to get rid of things because it can inactivate everything. And other things are the opposite. It's like I cannot inactivate anything, but I can inactivate it and I can modify the target. So each bacteria has its own things. So now the other thing we did is like, is that true? I mean, can we just map something to card and that's it? So we just do more and more experiments. So Gerardo did all the experiments for every strain with every antibiotic to see if this was, again, if this was true. So he found, again, that every time he saw something in card, it really matched, the reality. These things it's really working. Let me show you. And then these things, they really have, enzymes to inactivate the antibiotic. These enzymes are called beta-lactamases. These beta-lactamases are classified as A, D, and E, and we have them everywhere. But here, look at this. Class D is mostly in the antagonist. Class A and B are in the resistant. So they have different strategies. Again, the enzymes are not everywhere. There's not a lot of horizontal gene transfer. And you can see that there's been an evolution within each taxonomic group to become specialized in the things they use as antibiotics and in the things they use to inactivate. And so I'm not going to tell you the details, but what if these are not really beta-lactamases? I mean, this can just look like beta-lactamases. But Ivan actually did the experiments. We know there are beta-lactamases. And these beta-lactamases have different characteristics, and some resist clavulanic or EDTA or whatever. So Ivan has analyzed every beta-lactamase, and we know exactly what it does. And not just that. If you take, let me show you, if you look at beta-lactamase and see how much it is made by the antagonist strain, you have this thing. And how much by the resistant, you have this. But if you put together the resistant and the sensitive and the other one, it goes down. So it seems that you don't need to make more of the antidote when they are together. In fact, what we think is that you are limiting the synthesis of antibiotics. So those are the kinds of things we are studying, and we now think we have to do the couple, the resistant, for just from clinical relevance, and we have a very interesting model to understand, other things. We are finding things that do not match what we see in the clinic, so we hope we can show that, it's very complex. You don't need the pressure in the clinic, and you can stabilize this thing, and that's what this higher order interaction means. And we have these emergent properties where everything we know about beta-lactamase doesn't make sense, about antibiotic, it doesn't make sense. So we can uncover new things by putting them together. And so basically, see, I already acknowledged all my people and I'm done.